Hello students, so in this video we will be understanding about DNA fingerprinting or DNA profiling. Now students, it is said that 99.9% of the DNA is same in all the human beings and 0.1% of the DNA is different, that is shows polymorphism, and this 0.1% difference makes each individual unique. DNA fingerprinting is the identification of the difference in this 0.1% DNA sequence. Now, when we divide the complete DNA on the basis of density, we get two peaks. The larger peak is known as the bulk DNA, which is the 99.9% part of the DNA. It is the coding sequence. And the smaller peak is the satellite DNA, which is different in every individual and is a non-coding sequence or junk DNA. The satellite DNA is divided into two parts based on the length: the mini satellite DNA and the microsatellite DNA. The mini satellite DNA is also known as VNTR, that is variable number of tandem repeats, and microsatellite is also known as STR, that is short tandem repeats. The mini satellite or VNTR is the one which is used for DNA fingerprinting. Satellite DNA are repeated sequence DNA, that is, this part of DNA consists of repeated sequence of nucleotides. For example, ATGC, ATGC, ATGC. Now, the length of this sequence will be different in different individual, and in the same individual different in every cell and chromosome. Now students, we know we have 23 pair of chromosomes, that is a total of 46 chromosomes, and these repeated sequence or satellite DNA are different in each of these chromosomes. Now, for example, let us consider only the chromosome number 1 of yours and mine DNA. Now students, let blue one be your chromosome and the green one be mine chromosome. Now, the first chromosome of the homologous pair will have the satellite DNA ATGC repeated for 6 times, and the second chromosome of the pair will have the satellite DNA ATGC repeated for 3 times. Similarly, for my chromosome, the first chromosome of the pair will have the satellite DNA ATGC repeated for 2 times, and the second chromosome of the pair will have the satellite DNA ATGC repeated for 7 times. Now, how do we know this? How do we know the difference between the VNTR sequences of your chromosome and my chromosome? We know this by DNA fingerprinting methods. The various methods used in DNA fingerprinting to find the VNTR sequences includes isolation of DNA, centrifugation, digestion of DNA using restriction enzyme, electrophoresis, sudden blotting, hybridization, and autoreplication. radiography. Now, in the first step, that is isolation of DNA, the DNA will be separated out from the cell. So, your DNA and my DNA will be separated out from our respective cell. Now, we will allow our DNA to undergo centrifugation, where the satellite DNA will be separated out from the rest of the DNA. And the satellite DNA will be used for the DNA fingerprinting. Why? Because satellite DNA or VNTR is the sequence of DNA which is different in all human beings, and our motive is to find out this difference. Now, after centrifugation, we got our own VNTR, that is your VNTR and mine VNTR. Now we will allow our VNTR to undergo digestion, that is cut down into small fragments with the help of an enzyme known as restriction endonucleases. This enzyme, restriction endonucleases, acts as a scissor, and hence the enzyme is also known as molecular scissors. Now, after this process, we will get the fragments of your VNTR and my VNTR.